So for today's lecture, let us start with the introduction portion of your forensic microscopy, different types of microscope. So in this, basically, uh, the objective of this lecture will be like, once we end with this topic, you should be able to understand what are the different parts of the microscopes and how does the light travels inside the microscope. That is one thing. Then what are the different parts? How does the light travel? What are the different types of microscopes and how do they work? What is the principle? What is the, how does the reaction or the interaction of light and sample is taking place so that we are getting a visible result? Okay. So those things you will be able to understand in the coming classes. So so everybody might have seen the microscopes in your laboratory you might be having, or in your schools you might have seen the microscope. So as we see, like a general microscope, every microscope will have like three basic parts. That is like the three basic parts of your microscope will compose of like the head portion and the base portion and the arm portion. So three different parts of any microscope, whether it is a stereo, compound. So in that we have like the head portion will be the place where we keep our eye and through which we are seeing. That is one thing, the head portion, which will also include the objective lens and focusing lens. Okay, so that is, there are different components also that we'll see in the previous slide. The base of any microscope, which gives a support to the microscope. So actually it is composed of all the setup of the microscope. So whether it is the arm portion or the head portion, they are all standing on the base. And the base is also having a source of light which focuses on the sample. So that also we'll see. Then we have the middle portion of the microscopes, that is arm portion. So we can say the arm portion is the place of any microscope which connects two parts of the microscope, that is head portion and the base portion. That is one thing. And it also helps you to carry the microscope. Okay. So these are the three different parts of your camera, microscope. Then one more thing is like, how do you carry the microscope whenever you are handling any microscope? So first thing is like the microscopes at times they are like the normal stereo microscopes or the compound microscopes. They'll be costing around 40,000, 60,000. So it is a very costly instrument, right? So every instrument you should be able to like handle it properly. So suppose if you have to shift the microscope from one place to the other place. So what you do is, as I told you, like from the arm portion, you can pick it up and you can transfer that thing. Then the thing is like, suppose the arm portion for compound microscope is there. So compound microscope, obviously at times it is very heavy and costly also. So whenever you are holding it from the arm side and you are picking it up from the surface, at the same time you put your another hand under the base of that microscope for the safety reason, because even if you slip that thing or accidentally you lose a grip over that, so there should be a support for that. Okay, so it is like a care for the instrument. So whenever you are handling such kind of instrument, so make sure that your base, your one hand is at the base of the microscope and one hand you are holding the middle portion, the carrying portion. So that was about your small care thing. Then we'll just discuss about the different parts of the microscope. So you might have seen the microscope in your college and schools. So different portions or different parts of the microscope, as we can see that this portion is your eyepiece. Okay. So in the microscope, the optical part generally consists from this portion up to this area only. Okay. Important portion, that is the head portion. Okay, so what is the head portion consisting of? It is consisting of the eyepiece or the place where you are focusing or sorry, where you are viewing the sample, that is your eyepiece. Then you have your eyepiece tube. Okay, this tube can vary in length. It can be a short also, it can be a long one also. Okay, depending on the type of microscope that you are using. So next thing is about the body. Okay, this is your body. So this body, it holds the most important optical components of the microscopes. If you take the cross section of this microscope, you will see different types of optical instruments that are fitted in the what you call your head of this portion. Okay, so this will consist of different types of your lenses fitted into the head portion or the body portion. Then we have your revolving nose piece. So revolving nose piece, what it will do is like according to your requirement that you are going to see on the stage. So that thing you can just rotate your screen, rotate your this nose piece and fix your different types of lenses that are available. Okay. So these different types of lenses will vary in the different magnification also. So these are your objective lenses, maybe depending on the type of microscope that we are using. The objective lens will vary in 3 numbers or at times 4 numbers or 5 numbers. Okay. So here we can just rotate this piece and we can, according to our requirement, we can set up the magnification. That is one thing. Then we have the other part of the microscope, that is the stage portion. The stage portion is the place where you are going to keep the sample. So your sample will be placed here on the stage for viewing. Okay. So there is one, you can see this clips are there. So this clip will help you to fix your slide or to hold your slide on the stage. So once you put your this clips over the slide, so the slides will not be able to move. So now since your slide is fixed on the lens, on the stage, you can easily view your slide. Then another thing is there is a coarse and fine knob. So you can see this big portion, the big knob, that is your coarse knob. Then again we have this small part, this is your fine knob. Right? So what is the function of this actually? The function is to, whenever you are placing the sample on the stage, so whenever we place the sample on the stage, so we, with the help of this knob, we can move this stage up or down. Okay. So once you move the stage up or down, the calibration will be done, the focus or the sample that you are viewing through the eyepiece, whether it will be clear or it will be unclear. So that thing can be done with your coarse knob. So thing is, you adjust the magnification with your coarse knob. Then again, what the fine knob is doing. Once you have set your coarse knob, once you have set your focus of the sample, that means from the eyepiece, you are able to see the sample clearly. But still, you think like there is some small magnification still required. You can see the sample, but it is not very clear. Okay. So in that case, what you do is this knob, this fine knob will help you to make the sample very clear focus. So once you rotate this knob slowly, the sample that you have kept here in the focus, it will be much more clear. So a very good image will be formed. So this is the function of your knobs. Then base, of course, like it is holding the whole instrument. Then base is also having one important component, that is your illuminator. So some microscopes which are like using the source like halogen lamps, or at times this is absent. So instead of this, we can also have a mirror. So you might have seen those kind of cameras into your school where this particular thing is absent, and in case of this, there is a mirror placed so that we can use an external source of light and adjust the mirror in such a way that all the light falling on the mirror is entering into this diaphragm. Okay, it is focusing on the sample. So in this diagram, that mirror has been replaced by the illuminator. So most of the advanced instrument, advanced microscope, nowadays we use this illuminator only. Okay, so illuminator is actually it is having a bulb. So it is your tungsten bulb. So normal bulb, that is once you switch on this portion, so what will happen is the electricity is glowing this bulb, and this bulb is, after illuminating it, all the light is falling into this diaphragm. Okay, so there is a diaphragm which is going to limit your light. So because the sample what you are using here, uh, it may not require sufficient light or more light, or some samples may require less light or more light. So according to your sample, the requirement of light may vary. So how do you control that light? Here we are going to control that light with the help of this diaphragm. So we can rotate the diaphragm and we can limit the light that is coming from the illuminator and falling on the sample. Okay. So that is the function of your diaphragm. So again, there is one thing that is called condenser. Okay, just um, under this diaphragm. So diaphragm is limiting the light, and again the condenser, what it is doing is whatever amount of light is coming from the diaphragm, it is going to condense and focus on to this pinhole or the small hole. So just over that small hole, you are placing your slide, right? So all the light will be condensed, whatever is coming from the diaphragm, all those light will be condensed and it will come out of this hole and finally it will reach to the sample. So as we can see, this hole, we can give the name aperture because aperture is the place where your light is coming out ultimately and entering into the sample, right? So this is all about your this different types of different parts of your microscope and their function. Okay, so here what illuminator is doing is it is illuminating or it is a source of light. Diaphragm is limiting the amount of light that is illuminated by the halogen lamp. Condenser is condensing the light into one particular place, and that amount of light is coming out of this small hole that is called your aperture. Fine. So this is clear about your parts of microscope. Are you able to hear me?
Yes, sir.
Yes,
Yes,
sir.
sir.
Fine, fine. So I already told you like the three basic components of your or the parts of your microscope. That is your head, base, and arm. So now comes like what are the optical components? I have already told you in the figure itself, but we'll take some like the theory portion also. So optical components, there are two important components in whole the setup, in all the setup of your microscope. Two optical components are important. One is this and the other is this one. Okay. So optical instrument or optical component important is this one and this one. If you vary this thing and this thing, the whole image, the whole calculation or calibration will change. So your eyepiece, generally your microscopes, once they are supplied with the basic type of the eyepiece, and the magnification for those eyepiece will be either 5x or generally 10x. Okay, so whenever we purchase the instrument will have both, like it can be supplied with the 5x also, and in addition to 5x magnification, we can get 10x also. So I, sorry. So eyepiece is what you look through at the top of the microscope. Typically, standard eyepieces will have a magnifying power of 10x or 5x. Optional eyepieces of varying powers are available, typically from 5x to 30x. So that is about your eyepiece. Then second is the just below that eyepiece, the tube portion. So the eyepiece tube holds the eyepieces in place above the objective lens. Binocular microscope heads typically incorporate a diopter adjustment ring that allows for the possible inconsistencies of our eyesight in one or both eyes. The monocular, that is one ocular microscope, does not need a diopter like this instrument what you saw in the in the previous slide, in the first slide, that is monocular. That means only one single eye you can see. So in our labs you might have seen like the microscopes are having two ocular lenses, two with the help of your two eyes you can see. So that is the not monocular but a binocular. Okay. So mono means one, bi means two. So obviously you can understand the meaning of this. Okay. So what is objective lens? The bottom portion of your, just above the stage, the objective lens through which you are going to view the sample, which you are going to focus on the sample. They are the primary optical lenses on a microscope. They range from 4x to 100x and typically include 3, 4 or 5 lenses depending on the type of the microscope that we are using. Then we have the nose piece. So nose piece, actually what it does is the it houses or it holds the objective lenses. The objectives are exposed and are mounted on a rotating platform so that different objectives can be conveniently selected. Standard objectives include 4x, 10x, 40x and 100x. And apart from this, this 100x can also be supplied with the oil immersion objective lens. So that is one different kind of lens that we'll talk later. What is the oil immersion? So for now, suppose we have 4x, 10x, 40x and 100x type of your objective lenses. So they have a different power objectives. Okay, so this is what we are talking about, the nose piece. Okay, so this is a rotating device. We can just rotate whether we want 10x, whether we want 40x, whether we want 100x. We can just rotate this and according to our requirement, we can focus on the sample. So then we have the coarse and fine focus knobs that are used to focus the microscope. Increasingly, they are coaxial knobs. Coaxial means they are into the same plane. That is to say, they are built on the same axis with the fine focus knobs on the outside. Coaxial focus knobs are more convenient since viewer does not have to grope for a different knob. So what it means to say is the coarse and fine knobs, they are placed in one plane only, so that while you are adjusting the magnification, so first thing is we should start with the coarse knob. So once we are operating the coarse knob and we have got the good magnification, good view is obtained through our eyepiece. And still if we want to magnify that thing, so what we do is we adjust with the fine knob. Okay, so that is what it is saying. Then we have the stage, obviously the place or the platform where the sample is kept or where you keep your slides. So that is your stage. Then you have the stage clips. The stage clips will help you to hold the sample or to hold the slide. Okay. So at times you can like operate the stage manually also. you can shift the stage left to right, forward, backward also. So some cameras, some microscopes, they have this function also. But some microscopes, they have a fixed stage. Some microscopes, they have a rotating or moving stage. So the advantage of moving stage is like, if you see this stage is like fixed, it is not going left or right, forward and backward. Okay, this will be fixed. So what you have to do is you will have to manually adjust your slide on this. So you have to move your slide and then only you can see the sample through this. But the advantage of this moving stage is like, suppose if you have kept your slide here and given the stage clips, that means the sample is now fixed on the stage. So now you don't need to touch the slide. You don't need to touch the stage. There are some rotating knobs just below this stage, and with that knob, either you can move your stage forward, backward, left or right. Okay, so that is the advantage. So you don't have to touch your sample. Just fix your sample on the slide once, and with the help of the rotating knob, you can just shift your stage according to your convenience. Then you have your stage clips, which helps you to hold the evidence or your slides. Then you have the aperture, that is the hole in the stage through which the light reaches the stage. Illuminator is the light source for microscope, typically located in the base of the microscope. Most light microscopes use a low voltage halogen lamps with continuous variable lighting control located within the base. Okay, then there is one more thing, like, this illuminator, whatever amount of light it is giving, that amount of light also it can be controlled. That can be controlled. Okay, so that is what it is saying. With continuous variable lightning, it can be controlled also within the base. So in this diagram, in that picture, you will not be able to see because the variation was not there. But in second image, I will show you like how it is controlled. Then you have your condenser. I already told you the amount of light which is coming, it will be condensed or it will be focused into one small place. Okay. Then you have your iris or diaphragm. Okay. The amount of light reaching the specimen is located above the condenser and below the stage. Most high quality microscopes include, this is one company name, okay, Abbey condenser. Within iris diaphragm combined, they control both the focus and the quantity of light applied to the specimen. That is what I am saying. They control both the focus and the quantity of light applied to the specimen. That means whatever the amount of light is coming, that is to be controlled, and then the focus, where it has to go, whatever the amount of light it is coming, where it should be go, whether it should be focused into one plane, one small area, or it should be given in a large area. Okay, so that thing is also controlled. Then you have your condenser focus knob, move the condenser up or down to control the lightning focus on the specimen. So this is about the optics, the light traveling. This is how the light is traveling inside the microscope. Okay, so this is the place. So this was about your illuminator or the halogen lamp or the light source. Okay, so from this light source, the light will travel through the condenser. It is coming out of the aperture on the specimen. The light will pass through the specimen and it will reach to your what is your objective lens. Okay, so whether it is 5x, 10x, 100x, so the light will just enter into that objective, and again after entering into the objective, the light will pass through the projector lens and reach to the eyepiece. So eyepiece also different magnification we are having. 5x is we can have, or we can have 10x also. Okay. So once, are you able to hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Oh, just hold on for some
Yes,
more
sir.
time. Okay, okay. Hold on for some more time because time is running out. We'll just end this session.
Okay.
So the
Okay.
light is finally reaching to your eye. Outside this eyepiece, we are viewing. Okay. So finally, how the light is traveling is from the illuminator, that is your halogen source, through the condenser, through the aperture, throughout within the sample, the light is focused, reaching to your objective lens, which varies into different magnification. It is projected through the projected lens into the eyepiece. The eyepiece may also vary whether it is 5x or 10x. And finally, we will be able to see our sample, what is placed on the stage with the eye. Okay, so that is how the light is traveling inside the microscope, right? So depending on the traveling of light within the specimen, okay, so whether the light is traveling within the specimen or how the light is affecting the sample or how the light is interacting with your specimen. Okay, so whether the light which is traveling here, whether it is absorbed or deflected or reflected by this specimen, or whether the light is traveling through the sample. Okay, so depending on that, we have like different names, that is dark field microscopy and bright field microscopy. Okay, that we will take in the next class. Okay, so for today, just remember like how the light is traveling inside the microscope, what are the different parts of the microscope, and what is their function, and how does the microscope work. That is the basic. So for today, we'll end with this thing, and later on we'll continue uh, with your dark field and bright field microscopy. Okay. So for today, thank you all.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir. Hello? Hello?
Thank you, sir.
Hello?
Yeah, tell me.
Hello, Amare.
Okay, okay. Whoever was present today, they should share your presentations in our group. Okay?
Yes, miss.
Okay,
So,
miss.
so others can refer it. Hello?